everybody. Well, we've talked about a lot of things in this series on coping with COVID, and this time I thought it would be interesting, since two of the vaccines are out now, but there's at least three more likely to come out in the not too distant future, that we might want to just take some time and talk about how these vaccines have been developed in this very short period of time, less than a year really. And frankly, to me at least, it's awe-inspiring. It's awe-inspiring that people have been able to figure out how to make a vaccine, starting from scratch really, and starting at this time last year, and then doing the testing, getting these huge populations of people, of volunteers, to get either the placebo or the vaccine, and then get the data put together, get it before the FDA, get it approved for emergency use. So let's go through a little bit. And remember that the coronavirus, big circle with these spikes that stick out. In fact, it's called corona for crown because of the spikes. And those spikes are what attaches the virus to the cells of your body. And so the idea has been to make an antibody to the tips of those spikes. And once that antibody is made, then the antibody will coat the spikes. And you see that in this third image here, which, by the way, I took from Regeneron's website. Hope they don't mind. So that's the basic principle of all vaccines. And all the vaccines against the coronavirus that are coming out are all made to attach to those spikes. So there's three kinds. There's what we might call the genetic vaccines. These are the messenger RNA vaccines. Second kind is what might be called the viral vector vaccines. I'm going to go through all these in a moment. And the third are what we'll call protein-based vaccines. So three different types. So let's start with the genetic type. There's two companies that you're probably very familiar with now, Pfizer and BioNTech, and Moderna. These are the two companies that have come out and gotten emergency use authorization already for their messenger RNA-based vaccines. So let's look what's the basic concept. Messenger RNA is the substance that travels from the DNA into the cell and tells the cell that what it should manufacture, what kind of protein it should make. So there's a messenger RNA for every type of protein. There's a messenger RNA for, say, hemoglobin, a messenger RNA for the enzymes that our intestines secrete to digest our food. So it's a very, very normal part of the body's mechanisms. But what's been done here is to synthetically create a messenger RNA that is the messenger RNA for that spike protein. So it was able to do it because they know what the genetic sequence of the virus is. And knowing that genetic sequence, they could say, okay, this is the piece for the spike protein. We'll make a messenger RNA which will send that message. So that's what's been done. That's the real basis of these messenger RNA vaccines. So I show you some diagrams here, and by the way, these all come from the New York Times. You see the messenger, that's in red. It looks like a wavy little thing, and then around it is sort of a purple colored thing. And that's a lipid particle. It's a lipid that sort of covers up the messenger RNA so that when you get an injection, the messenger RNA doesn't get destroyed by various substances in your body, which would quickly destroy it if it had the opportunity. So it's covered with this lipid, and that gets into your cells, into the immune cells of your body, and so it will then make the spike protein. And I show here a messenger RNA in a nanoparticle, and it's just entering into the cell, and that's where the blue arrow is there. And then once the messenger RNA is in there, it turns on the cellular machinery and starts making those spikes. And you see here, now look at the green arrow down at the base, those spikes have been made, and they assemble around the bottom of the cell. Actually, they assemble all the way around, but shown here is just at the bottom. So now they're on the cell. Now your body sees that and says, "Hey, that's foreign. That's not normal," and so it starts to make antibody to it. And so once you have the antibody, then the antibody can come back and coat those spikes on the real virus should you come in contact with it, kill it, destroy it, or as they talk about in science, neutralize it. Neutralize it. So that's the basic concept. So let's go back again to this time last year. That's when China published the sequence, the genetic sequence of the entire virus. Very quickly, people at BioNTech and at Moderna, by the way, they know each other, they've been working together occasionally, but they both saw that and said, wow, we could make a vaccine. So let's talk about BioNTech first. It's a small German company. It's got it had at the time about 3,000 employees. The two co-founders, husband and wife, they saw that and said, we're going to do this. And the one of the two sort of sat down at his computer that day and designed the messenger RNA. Said, we can make this. Went into the company the next day, got all 300 people together and said, stop everything, stop everything. They've been working on messenger RNA technology for 20 years, so it's not new. 20 years, they had a lot of stuff in their pipeline, but they said, everything's going to stop, we're going to make this vaccine. And a lot of people were about to go off on, it was this time of year, wintertime, they were going to go off on holiday and go skiing, and he said, please don't, let's just come to work. We'll work 6-7 days a week, we are going to do this. And they did, and very shortly they had they did a couple, actually had 20 different candidate vaccines, messenger RNAs, but they narrowed it down to 2, tested those in humans, and one seemed to be better than the other, so that was, they nailed it down to that. But then they said, look, we're a small company. We need a big company that knows how to take this and do the studies. We're going to have tens of thousands of people vaccinated. And so we're going to need a company that can manufacture this stuff and do the studies and then be able to manufacture it in, millions and millions and millions of doses. So they went to Pfizer. They had a relationship there anyway. The head of their vaccine group said, okay, we'll go. And she went to the CEO and he agreed. And he really agreed. And he said, okay, I want this vaccine out, done, and approved or authorized in October. They all said, I don't think so. But in the end, they did, and they put it into 43,000 volunteers, well, half and half placebo, half the vaccine. And by mid to late November, they had the results, went to the FDA, and by December the 11th, it was approved. It was, I shouldn't say approved, it was authorized for emergency use, EUA, emergency use authorization. That was the first one. So now let's go to Moderna. Moderna, I did show it in red, the last 3 letters of Moderna are RNA. This is a company that's been around since 2010. And they're only focused on using messenger RNA technology for vaccines. A lot of it for cancer, a lot of it for infectious disease. And they actually have 12 different candidates in the pipeline right now in clinical trials. So again, my point being, we think of this as new. It is new, but it's not like just yesterday. These companies have been working on this technology for a decade in one case, a couple of decades in another. So it's just important to understand that these vaccines are new, but the technology's been around. In the case of Moderna, well, just like BioNTech, the folks there saw the genetic sequence that came out from China. They said, hey, we can do this, we can make this. They got right to it. And they had a relationship with the National Institute of Allergy and Infectious Diseases. So they were working together with them and with another group in the government. And they were able to move things along quickly. They got some outside funding and some funding from the government. And they've been able to carry this forward. They got their studies underway. They were actually running neck and neck with Pfizer-BioNTech. But then they got sort of stalled for about 3 weeks while there were some differences of opinion between these various agencies as to how you were going to get the appropriate spread of different ethnicities and gender and ages into the studies. Well, anyway, they finally got over that. And again, by late November, they had data, and on December the 18th, theirs was approved for emergency use. So there we are. A couple of extra points to point out. I already said, yes, these are new vaccines, but it's not a new technology. Second thing, which I found kind of interesting as I was researching this show, is there's a lot of immigrants involved here. The two co-founders of BioNTech were Turkish immigrants to Germany. The head of vaccine development at Pfizer was a German immigrant to America. The CEO of Pfizer is a Greek immigrant to America. And we'll come to Novavax in a little while. One of the key scientists there is an Indian immigrant that came to this country years ago, and has been sort of the key person in developing that vaccine. But let's go on now. Oh, one last thing. I have heard people say, I'm worried about this messenger RNA. It's going to get into my DNA. What's it going to do? It doesn't. It can't, actually, because it is a messenger RNA, and once it's in the cell, it does its job. It's destroyed. It's done. It's gone. It can't get into your DNA. This doesn't work that way. That's just the way I'll say it. So let's look at the viral vector vaccines. A different technology, but with the same endpoint, to get those spike proteins created and then recognized by our immune system and an antibody made. So the two companies that are working on that are relevant because their vaccines may get approved very soon. First one is Oxford University. It's tied up with AstraZeneca, and the other is Johnson & Johnson. So again, both companies have been doing this type of work for years, so it's not, again, a new thing. But the whole concept here is this. They take a modified cold virus, an adenovirus, a modified cold virus that can get into a cell but can't replicate. So it gets in, but then it's done. And what they do is they put into that the gene for the spike protein. So this virus is then carrying the spike protein gene into a cell. Here's some videos, not videos, images to help you see this through. So we'll start off with the first one. Is we've got the virus. You can see the spikes around it, and its DNA is on the inside. So they extract the DNA for just the spike protein. They take that, they put it into this adenovirus, which you see there as a hexagon, and that then virus can get into the cell. And you see that in the next one. I show it with a blue arrow coming in from the left side at the top. The virus is getting into the cell. Now the DNA comes out. It creates messenger RNA. The messenger RNA starts to make directs the making of the spike protein. And again, what you see here with the green arrow in this next one is there are those spikes on the bottom. And now once they're there, the body recognizes that, says that's foreign, it shouldn't be there, will make antibody. And so that gets made, and there you have it. It's going to grab any coronavirus that comes by. So that's the essence of the concept of viral vectors. Again, you might be concerned and say, well, wait a minute, that's DNA that you're putting into the adenovirus, but it can't go anywhere. It can't do anything. It can't integrate into your DNA. It's just going to get broken down within just a very short period of time inside that cell. So let's just talk about Oxford University, AstraZeneca. The Oxford people had developed this. They actually had some relationships with other companies, but somehow that didn't work out. And they went to AstraZeneca, the same thing. They needed a large company that knew how to put this stuff together, how to run huge, huge trials, which they did. And it has now been approved in the United Kingdom. AstraZeneca being a British company, and Oxford, obviously, Oxford University. It should be, there's also a huge study going on in the United States. I think it's 30,000 again, 15,000 placebo versus vaccine. And those results should be available within, maybe by the time you're watching this TV program. Anyway, it'll be quite soon. We'll see what the results are. The results they had from England were a little bit confusing, but certainly it was working at least 60 to 70% of the time, and maybe 90% of the time. So we'll see how it matches up against the Moderna vaccine and the Pfizer BioNTech vaccines. Now Johnson & Johnson, they're using exactly the same technology. You know that company, everything from Band-Aids to surgical implants, hip implants, and so on. But they have a large vaccine development group. And so they've been working on this, the same thing, very large trials going on. And the expectation is that that data will be available come the first week of February, or certainly the second week of February. So again, by the time you see this program, it may well be that that data is out. And once it's out, the FDA will get it. And if it's good and everything falls out okay, they'll again issue that emergency use authorization. So it's exciting. If that's all happens, that would be 4 approved vaccines. So let's now go on to the protein-based vaccines. And the company here is Novavax. Novavax is a small company, Gaithersburg, Maryland. Actually, it's kind of interesting. Although they've been working on this for these types of things for some time, their stock price had really dropped way, 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 way down. And they were even going to possibly be delisted from the exchanges because their price was that low. But then all of a sudden came a pandemic. And they saw this and they said, we have the technology, we can do this, but they're going to take a different approach. And they said, we're going to make that spike protein directly, and we'll inject that. We won't go through all these other steps. We'll do the steps outside, and then we'll just inject the spike protein. So here's how that works. They start off with the DNA of the gene for the spike protein. And this time they insert into a virus called the baculovirus. Honestly, I never heard of baculovirus before, but it's been worked with before for this sort of thing, in the baculovirus. And the baculovirus they then use to infect the cell of a moth. Okay, you're going to wonder why a moth. Anyway, it works. They've been doing this for years. This technology has been around for over 20 years. So they get the DNA into the moth cell, and you see that here. I got a blue arrow. That's the baculovirus with the DNA in it. It goes into the moth cell. The moth cell now starts to make messenger RNA. From there, the messenger RNA starts to make the spike proteins. And this time, remember, it's not being done in you, it's being done in this moth cell. So what they're able to do is they make loads and loads and loads of spike proteins, and they extract them out, purify it, get rid of everything else. Now they have the spike proteins, and they add it to a carrier, again a nanoparticle, and it's 14 of these spikes around this one particle. And that's what they inject into people as the vaccine. A little oversimplified, but it will do. So again, they add an adjuvant to it because they know that if they add the adjuvant, it works a little bit better. But anyway, they end up with the neutralizing antibody, which again you see the picture here of the coronavirus. If you come in contact with it, these antibodies are going to just coat the whole thing just like crazy, and again neutralize it and get rid of it. So that's this. Oh, and where are they with this? They're a little bit later in the whole process, but it's coming along. Their phase 3 studies have started, and again, you know, the very large 30 plus thousand people, and they'll probably not be finished though until into the spring, probably mid-spring. So we'll see what happens with that. But anyway, it's coming along. So that would be the 5th vaccine. And there are others out there. Merck, a huge company that's been a world leader in vaccine development, they had 2 concepts that they were working on, and neither of them worked out. So they've actually withdrawn entirely from making a vaccine. It just shows why it's really good for us that so many different companies decided to work on developing these vaccines, because here's a big company, Merck, who you know, this is one of their big businesses. They understand this, and yet they had 2 failures. So again, it's a credit, I think, to some very ingenious people and big companies that were willing to commit. I think I mentioned before that Pfizer committed literally $1 billion to make this happen, where the CEO who said, I want it in October. The other companies did essentially the same thing. And they also got a commitment from the US government, each one to make a million doses, and they got some payments up front to help cover the costs going forward. So they were covered just in case it was a failure. And that was part of the whole idea of the Operation Warp Speed. We may have some failures along the way, and if we do, then we've got some backup things. It looks like we may have 5 vaccines. And the Johnson & Johnson doesn't require a deep freeze, neither does the AstraZeneca. It requires freezing, but not a deep freeze. Very deep freeze, and the Novavax can be stored anywhere, so it could be used in a lot of developing countries, for example. So those are the various ones. Again, I think it's really awe-inspiring that these have been pulled together like this in such a short period of time, and it's going to make a whole big difference for all of us in the days, weeks, and months to come. Thanks very much for watching.